So, this is for those of you claiming so confidently that the vaccine cannot possibly alter your DNA. I want to share some recently published research data from the Sidney Kimmel Medical School at Thomas Jefferson University, published June 11th, 2021, in the journal Science Advances. The scientists claim to have made a discovery identifying a highly efficient human reverse transcriptase that can write RNA sequences into DNA. In a discovery that challenges long held dogma in biology, researchers show that mammalian cells can convert RNA sequences back into DNA, a feature more common in viruses than eukaryotic cells. Cells contain machinery that duplicates DNA into a new set that goes into a newly formed cell. That same class of machines, called polymerases, also build RNA messages, which are like notes copied from the central DNA repository of recipes, so they can be read more efficiently into proteins. But polymerases were thought to only work in one direction, DNA into DNA or RNA. This prevents RNA messages from being rewritten back into the master recipe book of genomic DNA. Now, Thomas Jefferson University researchers provide evidence that RNA segments can be written back into DNA via a polymerase called theta, which could have wide implications affecting many fields of biology. This work opens the door to many other studies that will help us understand the significance of polymerases that can write RNA messages into DNA, says Richard Pomerantz, PhD, Associate Professor of Biochemistry and Molecular Biology at Thomas Jefferson University. That polymerase theta can do this with high efficiency raises many questions. For example, this finding suggests that RNA messages can be used as templates for repairing or rewriting genomic DNA.